And in this topic, we are going to study apoptosis. Did you know that every 40th second a person is committing suicide in the world? That is the sad reality, right? And what is more sad is a cell committing suicide. Cell is a very cutie thing, right? Very, very small. See this little guy, his name is Bittu, and he's a cell. And him committing suicide, right? Him committing suicide is a very, very disappointing thing. But that is not necessarily bad for obviously Bittu, but not necessarily bad for the body. It is a way in which we are clearing out, right? We are clearing out the things which are beyond repair, that is the cells, so that new cells can give its way. And this is very, very common, right? Obviously more common than human suicides. So let's see how it happens. Why does Bittu have to commit suicide? We'll study about that. See, there are two pathways, intrinsic pathway and extrinsic pathway. Intrinsic is from within, right? Within the cell itself, Bittu is going into depression and he knows that he is not good for society, so he is going to commit suicide. So that is the intrinsic pathway. Extrinsic pathway, someone outside is making him commit suicide, right? Someone is stimulating him. We'll go to there. First study the intrinsic pathway. Everything will start with mitochondria. Mitochondria is the energy powerhouse, right? We all know this. And that is the one which is responsible for apoptosis also. If it sees that there is not enough energy in the cell, right, it has a lot of injury and it is suffering, so better to take him out of misery, right? So that is how the mitochondria is going to function. That will cause cell injury, so that has some receptors, right? Obviously it has to sense something. So these are the sensors. These sensors are mainly BH3 only proteins, right? Remember this BH3, right? Behatar, that's how I remember. Behatar, I mean they want to improve the cells. So BH3 only proteins. So 3 Bs. Examples are mainly 3 Bs, that is mainly BIT, BEAM and BAD. 3 Bs, right? Apart from that, we have 2 more, that is Puma, Noxa. Right? Remember these are the examples of BH3 only proteins. And these are the ones that will increase the pro-apoptotic proteins, pro-apoptotic genes, and decrease the anti-apoptotic genes, right? Obviously, you want apoptosis to happen, right? That is the cell injury is happening and they want to happen. That's why increasing the pro-apoptotic genes. And everything will start with BA, not bad, but remember BA. So that is BAS and BAC. Whereas anti-apoptotic genes, everything will end with L. From L, remember, long life, right? So from L, so remember that is BCL, MCL, BCL, XL. See, everything is ending with L, right? And these are the ones, there will be decrease in the anti-apoptotic genes. So when that happens, the hero inside the mitochondria is getting activated, the weapon, the secret weapon, that is the cytochrome C. And what it does is that, as you can see here, he is going to combine with apoptosis activating factor 1. Remember 1, right? Apoptosis activating factor 1. And when he is going to get combined with him, these two guys can easily activate a cat. Remember, it's not a cat, but it is caspase 9. Remember 9? I want to remember a cat because cat has 9 lives, right? So act on 9th caspase, remember. It will act on the 9th initiator caspase. So that is for the intrinsic pathway. The common pathway, we'll come to that later, right? So just remember, till now the story, the intrinsic pathway, what it is doing, it is going to activate 9th caspase. That is the intrinsic pathway. Next, coming to the extrinsic pathway. See the external factor which I told you, right? That is nothing but this guy, that is tumor necrosis factor. And this tumor necrosis factor is a very fast one, right? Very fast. Remember, it's called as fast ligand because it will fastly attract with the fast. Fast is nothing but the tumor necrosis factor receptor, also called as CD95, right? That is a receptor CD95. It has that marker, that is CD95 marker will be there in the fast. And that will activate. This guy is going to go attach to the receptor and he is going to activate his special skill, the death domain, right? So that is, it will activate the fast associated death domain. And these obviously again are activating the caspases. And remember 9th was for the intrinsic, right? But here remember the Casper, right? So that is a Casper ghost. So here it is instead of 9th extrinsic pathway, right? So remember E for E, that is 8. So 8 and 10. 8 and 10 are the extrinsic pathway which is activated by this. And finally, so what happens in the end? In the end, this guy, who is this guy? He is looking like a death reaper, that is executioner. He's an executioner, he's going to execute you. So he is going to get activated, that is called as executioner caspase. In that the most common is 3 and 6, right? 3 and 6, the executioner caspase. So what he does, he's going to break the main skeletal framework, that is the protein, right? So protein breakdown happens over here, and also he is going to attack in the heart at depth, that is the what is the heart of a cell, that is nucleus, right? So there is again endonucleus activity over here. So after this, right, so we got the right debris over here, that is from the nucleus, that is endonucleus activity, and the protease activity, right, protein breakdown. So these things, they are going to get accumulated on a side, and a bleb like formation is happening over here, right? As you can see, that is nothing but an apoptotic body, and later it is removed from the cell, and they do something special. This apoptotic body, they do flipping of potass. What is this flipping? Potass is a phosphatidyl serine, and that is going to that is going to attract the big sister. Who is this big sister? I am talking about the macrophage, right? As you can see the diagram, macrophage. So macrophage is going to come and kill this apoptotic body. So as you can see, the cell is not entirely dead, but the majority of the waste is removed using the help of a macrophage. And just remember, Anne is a sister, right, like a macrophage. So this is stained by annexin. So when I'm talking about the stains, let's see the histology. So this is how an apoptotic cell will appear. The cell will become very small, right? Obviously, I mean, since apoptotic body and everything, it is giving a part of its body, right? So apoptotic cell will become very small. But remember, the cell outline is maintained and there is no inflammation. Remember, no neutrophils, nothing, not the white guy is not coming, so there is no inflammation. So when you look at in the gel electrophoresis, this is how it will appear. This is a normal cell, right? And this is where the necrosis is happening, and this is where the apoptosis is happening. In necrosis, because of the karyolexis, karyolysis, whatever we have studied, it will result in the smearing, right? Smearing can be there. Whereas in apoptosis, you can see step ladder pattern, right? Step ladder pattern is a very characteristic finding in DNA gel electrophoresis for apoptosis. So that is a step ladder pattern. Alright, now let's study something interesting. Necroptosis. What? Necropoptosis. Is it necrosis or is it apoptosis? This is a combination of both. You're like, oh my god, entire death, right? Dead God is here. So what happens exactly? It is a similar pathway, right? You're going to combine the two pathways over here. So extrinsic pathway we have studied, right, of apoptosis. The same thing was happening over here, that is increase in the tumor necrosis factor. FAS is getting activated, and FAS associated dead domain is getting activated, and then the caspase 810 is getting not activated, right? That is the catch over here. It is not going to activate the caspase, right, or the caspase, but instead is going to activate RIP, rest in peace, right? Not the caspase, but rest in peace 1/3. He doesn't want to deal with any ghosts, so rest in peace 1/3. Remember, that is the receptor associated kinase protein, and that, right, you want to rest in peace. So what do you need? You need peace, right? You don't need any energy, right? So there is low energy or ATP depletion. Basically, if you do a vigorous exercise, you will get the ATP depletion, and you can be rest in peace, right? You will sleep very well in that night. So remember the ATP depletion, and also you need oxygen, right? Fresh air. So remember, there is increased reactive oxygen species, that is oxygen reactive species, but this will stimulate the necrosis over here. So the necrosis is going to continue. So that is necroptosis. See, initially the pathway was all of apoptosis. In the end, it has cheated you, right? Caspase is sad now, right? Caspase was not activated, but instead RIP was stimulated, and that will result in necrosis. And where does it happen? It does not happen everywhere, right? And for this to happen, necrosis and apoptosis together, you need a very strong weapon, right? I mean very strong in the sense, I mean sharp weapon. You cannot create necrosis and apoptosis very similarly. We need a sharp weapon. From that, remember SARP. So it happens in steatohepatitis, right, and acute pancreatitis. And from R we have reperfusion syndrome, and P, remember brain somewhere, right? So that is Parkinson's disease. So this is necroptosis. And after necroptosis, we have something interesting, that is pyroptosis. What happens in pyroptosis? As you can see, that is the pyroptosis. There will be fever, right? Fever with apoptosis. It's not the fire in your belly, right? It's fever, right? That is the pyro for our body. So you cannot create fire, but you have fever with apoptosis. So what happens is that in viral infection, cell death will happen. Why? See, what is this? That is the banana peel, right? It looks like banana peel, that's what I made. So there will be decrease in the potassium and increased uric acid, because of which the cell death can happen, and that is going to activate Noddy. What Noddy? Do you remember Noddy, right? Our cartoon character. So from that, remember, it is going to activate Nod-like receptors in the cytoplasm, and they are going to activate the anger guys, that is inflammasomes, right? They easily getting inflamed and red and everything. So remember inflammasomes, and whenever inflammation is considered, it's all about leaking information, right? That is interleukins. So remember that is pro interleukin 1 is getting converted into interleukin 1, and that is going to activate C111, that is right, C111, and some places it is written as 4 and 6, right, complements. So when that happens, they are going to stimulate this apoptosis. So this is the entire process of pyroptosis. As you can see, viral infection, and that will decrease potassium and increase uric acid, and that is going to activate NOD or NOD-like receptors, and that is going to activate inflammasomes, and they are going to in turn activate the interleukins, and that the complements, and finally that will result in the apoptosis, right? So that is pyroptosis. Next we have ferroptosis. This is kind of easy. It is nothing but the increased intracellular iron. When that happens, there will be increased reactive oxygen species, right? This reactive oxygen species we have studied, right? It increases in necrosis, right? So that will increase cell death because of necrosis. Next we have autophagy. Autophagy, right? I mean cannibalism. That is what we can tell. Yes, it is true. Even though we are not cannibals, our cells are cannibals. They are going to eat other organelles. Okay, fine, that is okay. But eating self, yeah, they do it. You will be like, okay, fine, that is good for body. So when this did happen, just imagine that if I tell you are going to become a cannibal, you like, you cannot never imagine it. But if I tell you if you are dying of starvation, and if you don't eat someone, you are going to die. In that condition, right, you can become a cannibal. That can be still questionable, but anyway, remember starvation. And which cells are most starved? That is mainly the cancer cells, right? The cancer cells, they need a lot of nutrition, so they tend to cannibalize more, right? So remember cancer, and you don't really tend to eat other guys, right? But if you are eating, then there is something wrong with your brain. From the brain, remember neurodegenerative disorders. So let's see the autophagy. How does it happen? See here, we have the organelles, right? So these are the damaged organelles. And next, the first step is initiation, second step is elongation, and the third step is lysosomal attachment. We'll come to that. So first in the initiation complex and the nucleation complex are coming, and they're going to create a substance like banana-like substance, that is the plasma membrane-like structure near the organelles. And next we have light chain 3, that is going to get this, that is going to elongate this plasma membrane-like structure, and they'll cover it fully. This is what is called as phagosome. And after that, the lysosome will come and get attached to that. That is called as phagolysosome, right? That is the phagolysosome which you can see over here. So that is for the autophagy. Let's revise everything quickly from top to bottom what we have studied in right apoptosis. Apoptosis has two pathways, intrinsic and extrinsic pathway. Intrinsic pathway activates the CAT, that is mainly the caspase 9, whereas intrinsic factor is going to activate the fast associated death domain, and finally the 8 and 10 caspase. That these are initiator caspase, remember. Next, that is going to activate the executioner caspase, and that will give all the effects of protease and endonuclease. And finally, there is apoptotic body formation, which will be engulfed by the macrophage, and that is stained by annexin. And next is the famous DNA gel electrophoresis in apoptosis, that is stepladder pattern. And we have necroptosis, right? The combination of both does not occur in all the cells, needs special circumstances for that to happen. That is only seen in steatohepatitis, acute pancreatitis, reperfusion syndrome, and Parkinson syndrome. Next we have pyroptosis. Pyroptosis, as I told you, right? So this is because of fever that is going to increase, that is going to induce apoptosis. How? Mainly based on inflammation, right? Remember, inflammation is involved over here, but still it is activating apoptosis, right? But apoptosis in general does not have any inflammation. And ferroptosis, so excess iron is also not good. And autophagy, the famous cannibalism. So we have studied everything. So that completes our apoptosis. Thank you.